Today we start the chapter on inherited change, which is the chapter 16 of the syllabus, and it is for the A2 biology. Code is 9700. This is CIE, and this is the second video on this chapter. The previous one was on meiosis, and as we look at the syllabus, we start with 16.1a. Now it says explain what is meant by homologous pairs of chromosomes. Well, that's a very simple thing. Homologous pairs of chromosomes. Homologous pairs, as you know, all know that, like human beings have 23 pairs of chromosomes. So chromosomes are usually in pairs, and every species has a specific number. Now what is common about a homologous? Number one is the shape. Number two is the length. Number three is the position of the centromere. So where is the centromere placed? Now here you can see it's here. This is the centromere and this is the centromere. So they've got to be at the same position. So position of the centromere. And number four, they code for the same characteristic. Like for instance, if you have eye color here, so say this is eye color. It's very basic, and so you would have eye color here as well. Now please remember, when we're talking of chromosomes, we're talking of bases, and we're talking of the base sequence. So there would be a T here, a here, a T here, and an A here, and a G here, and a C here, and of course the phosphodiester bond. And this is the sugar phosphate backbone, and there's a sugar phosphate backbone on this side. And we have the G here, and a C here, and a C here. And in between these are the hydrogen bonds. There are 2 between A and T, and 3 between C and G. So this is what we remember from the AS level. But what we need to understand is, when I say eye color, it means that this is the gene for eye color, right? And this is the gene for eye color on the same. So if it's a pair of chromosomes, they code for the same characteristic. So if you've got eye color there, you've got eye color there. And then let's look at another feature, and that is, let's look at a blood group. So if blood group is somewhere here, and this is the gene for blood group, so then we're going to have the gene for blood group on this side as well. And this would be a pair. So you can't have blood group on one side and then maybe something else like hair color on one side. Well, you can't have that, but they code for the same character. That's why they're a pair, just like a pair of black shoes, and you have a buckle on one, so of course the other one must have the buckle as well. So it's a pair of shoes, a pair of socks. So homologous chromosomes are always in pairs. And what happens in DNA replication is that a copy of this is going to be made. So there's going to be another centromere. So we're going to have a copy of it here. And these two together will be called sister chromatids. Sister chromatid. So we have to understand is that now the two chromosomes, these were the original 1 and 2. Now the copy of 1, let's call that 3. And the copy of 2, let's call it 4. So 1 and 3 are the same genetic, the same base sequences. So if there is whatever the base sequence is on this side, the same base sequence is going to be on this side, because you've got to understand that it could be different alleles. We're just going to talk about that a little later. So homologous chromosomes, and then 1 and 3 will be sister chromatids, and 2 and 4 will be sister chromatids. Now the next thing that we need to understand is, as it says in the syllabus, explain the meaning of the terms haploid and diploid. And diploid we usually signify by writing 2n, and haploid would be just n. So the diploid number in humans, is 46, and the haploid number in this situation would be 23. So haploid means one of each pair. One of each pair. Like for instance, if we have 20 students in the class and there are 20 pairs of shoes, and so I say, okay, well, everybody takes off one shoe and puts it ahead, and if we just take one of each pair, and that would be the haploid number or the haploid. Now these, of course, haploid are absolutely essential. What we need to understand, they're essential when we are going to be forming the gametes. And the gametes are there because they are required for sexual reproduction. And the important thing is that, you see, the diploid number would be present in every cell of the human body. So every cell of the human body is diploid, right? Your skin cells are diploid, your stomach cells are diploid, your pancreas cells are diploid, your liver cells are diploid. So all the cells of the body are diploid. So, and this would be a pair of chromosomes, as we've just talked about that, a homologous pair of chromosomes. And then in DNA replication, of course, a copy of it will be made. So there's a copy of this one and there's a copy of this one. So the 46 chromosomes will become 92 chromatids. So this is something which you need to be very clear about, is that every body cell is diploid. Now, except what we have to understand is that where meiosis is going to take place. Meiosis is going to take place in humans, and that is called gametogenesis. So if it's a male, then in the male is going to take place in the testes, and that process is called spermatogenesis. Similarly, if it's a female, then it's going to take place in the ovary. So meiosis is only going to take place in the testes and in the ovary, and the process which takes place in the ovary is called oogenesis. So these are the two names that we need to be very familiar with and very comfortable using them and spelling them. So one is called spermatogenesis, and it makes sense because the testes the gamete produced is a sperm. And in oogenesis, the final cell which is formed, which is haploid, is called an ovum. Now the sperm is also haploid, so it's N, and the ovum is also haploid and it's N. Because what we have to understand is that when the sperm and the ovum fuse, now when the zygote is formed, this is the zygote. The zygote has to be 2N. And this is the beginning of every organism, every human being, every lizard, every monkey, every elephant. The zygote is the beginning of every living organism, even if it's a plant. The zygote is the beginning, and that's what mentioned in the Quran. From a drop, a human being was created, and that drop is the cell. The cell is the zygote. So the zygote is this one cell which is the beginning, and it contains the diploid number. So half of them, one of each pair, has come from the sperm, and that means it's come from the father, and one of each pair has come from the mother, and that is in the ovum. So the sperm and the ovum fuse to form a diploid zygote, and the sperm is haploid and the ovum is haploid. Now we can study the two different processes, and we have to know the names of it. So the cells, which are the initial cells, which are called the spermatogonium, which are of course in the testes. So in the testes what we have is the spermatogonium, and it divides by mitosis, and it forms the primary spermatocyte. So testes, spermatogonium, then primary spermatocyte is when it's going to mature and then it's going to start developing. Now we have to understand where meiosis 1 takes place. So primary spermatocyte and spermatogonium, these are still diploid. So these are still 2N and this is still 2N. Now meiosis takes place and the secondary spermatocytes are made, and these are N. This is N as well and this is N as well. So two cells are formed, and these are called secondary. You see, primary comes first, like primary school and then secondary school. So primary spermatocyte and then secondary spermatocyte, and then meiosis 2 takes place. So this was meiosis 1 and then it's meiosis 2. So meiosis 2 takes place and then we have 4 spermatids. Now of course these are all N. So number 1 spermatogonium, number 2 primary spermatocyte, number 3 secondary spermatocyte. So meiosis 1, after meiosis 1 secondary spermatocyte is made, and then meiosis 2 takes place and spermatids are made. And of course then the spermatozoans or the sperms are the ones which differentiate and form the final functional cell, and 4 cells are formed. So we've got to understand this is what is happening in the testes. Now if you look at on the other side, what is happening in the ovaries? So in the ovaries what we need to understand is a slightly different phenomena. Well, it's quite different. The initial cells are called oogonium. So spermatogonia or spermatogonium and oogonium. And then of course it grows and matures, and this is of course, please remember here mitosis is taking place. And then what is formed is a primary oocyte. So primary spermatocyte, primary oocyte. So we have to be very clear about that, is what is what. So we have primary spermatocyte was in the testes, in the ovary we have the primary oocyte. Then meiosis 1 takes place. And now we have something which is a little different than what is happening in the testes, is one cell is larger, there is unequal division of the cytoplasm, and the secondary oocyte is bigger as compared to the second cell, which is this first polar body. Now this of course will die and disintegrate, so it's a non-functional cell. And then we have meiosis 2 taking place. And again there's unequal, and this is called an ootrade, or its final thing is the ovum which is going to be formed. So the ovum, I mean this is not very important to remember, but this is what we need to understand, is that this is going to finally form the ovum, which is only one functional cell. So as opposed to 4 functional cells, there is only one functional cell, and the second polar body is formed. So you see, both these polar bodies are no good to us, and they just die and disintegrate. So the final cell is only one ovum, and it's produced once a month. So a woman produces this ovum once a month, while the man produces sperms all throughout and every day, and is slightly different than the main processes. So in the testes, the process is 4 sperms being formed, and in the ovum in the ovaries we have 1 ovum. So in the ovaries what we have is 1 ovum, which is a functional cell, and this is once a month. So once a month we have an ovum being released, and then of course we talk about how it happens during which part of the menstrual cycle. So this is what we need to understand about the gametogenesis in humans, and now we have to study how this happens in plants. Now what we need to study is the formation of pollen grains, and if you look at the syllabus, it just you have to understand how again gametes are being formed. The pollen grains are the male gametes. So inside the anthers you have the diploid pollen mother cell, and this is going to divide by meiosis. So meiosis 1 is going to take place and meiosis 2 is going to take place, and we're going to have 4 haploid cells. So now these are all haploid. 4 haploid cells have been formed. Now the nuclei of each of these haploid cells divides by mitosis. So there is no cytokinesis. So we still have the 4 cells. But now what has happened is that the nucleus had divided, so we have now 1 cell but with 2 nucleuses, and both of these are N because it's only nuclear division. So the cell itself does not divide. So now we have 1 cell with 2 haploid nucleuses, Now these cells mature into pollen grains. So you can see here what has happened is now they have matured into pollen grains. So we had the pollen mother cell, which was 2n, then 4 haploid cells, so meiosis 1 and 2 has taken place. Then we have just mitosis, and you have young pollen grains containing 2 haploid nuclei, and then of course you have the mature pollen grain. Now this whole thing is happening inside anthers. The filament is just the stem which the xylem and the phloem pass through it and provide it water and mineral salts, and also take away the nutrients or bring it the nutrients. So this is what we need to understand about the formation of pollen grains. Now we have to understand what happens inside if you remember the female part of the flower, there's the stigma, then there's the style, and then there's an ovary, and inside the ovary is an ovule. And inside the ovule is a large diploid spore mother cell. Now this cell divides by meiosis. So we're going to have meiosis 1 and we are going to have meiosis 2. So meiosis 1 is going to take place and meiosis 2 is going to take place. Now once meiosis 1 and 2 have taken place, what are we going to have? We're going to have 4 haploid cells. So then we've got 1, 2, 3, 4. Now all these cells are haploid. So the cells divide by meiosis and produce 4 haploid cells. All of these degenerate. 3 of them degenerate. And the one surviving haploid cell, this surviving haploid cell, this is the one which has survived, now will develop into an embryo sac. So once again, large diploid spore mother cell develops, cells divide by meiosis, meiosis 1 takes place, meiosis 2 takes place, and we have 1, 2, 3, 4 haploid cells. 3 of these degenerate and 1 cell is left behind, and this is now going to develop. Now this embryo sac grows bigger, larger, and then it follows a very interesting pattern that is going to start dividing by mitosis. Now as you can see here again, this is the 1 cell divided by mitosis, 4 cells formed. Now we can see here this is that 1 cell, and it divided by meiosis, 4 cells formed. Now 3 of these die, and 1 cell now starts to divide by mitosis. So we have, please remember, no cell division takes place. So now there's 1 cell but with 2 nucleuses. And then mitosis takes place once, then mitosis takes place again, and then mitosis takes place again. So we have 3 times mitosis. So at the end of that, what do we have? We have a cell with 8 nucleuses, with 8 nuclei. And in this cell, what is going to happen now? First we had the original cell which had 2n or n. The original cell here had 2n, and then we have every cell has n in it. So now we have n nucleuses. So we have got 8 nuclei now. 3 goes in the north pole and 3 go at the other pole. So we have 3 here and 3 here, and then there's another one here, so 4. So we have 8 nucleuses. Now one of these, as you can see here, one of these is called the egg cell or the female gamete. So as you can see the label here, this is the egg cell or the gamete. So this is the one which is the egg cell and the gamete. So it's only this cell which is going to be the functional cell, the functional nucleus. The rest of all these are going to not much of a use, but I mean we're not going to study endosperm in which two of these nuclei fuse for another one to form the endosperm nucleus. So this is the embryo sac, larger haploid nucleus divided by mitosis 3 times, forming 8 haploid nuclei. And one of these, now this one is the female gamete. Now at fertilization, this has to fuse with the male gamete nucleus, which is coming in the pollen grain. So as you know, in plants, what do we have? First we have pollination, which is transfer of pollen, and then we have fertilization. And after fertilization is then fruit and seed formation. So now I'm sure you can understand it a little better, that what is happening. So what is happening to form the female gamete, and what is happening to form the male gamete, and then of course they fuse at fertilization. So first pollination, in which the transfer of pollen has to take place, and then the fusion of the male and the female nuclei. Now let's look at some definitions, which is of course part of 16.2a. The first one is gene. What is a gene? Gene is a short length of DNA. It is a short length of the chromosome. So a chromosome will have many genes on it. So short length of DNA which codes for a polypeptide, or you can say a protein. But it also, number one, codes for a polypeptide, and secondly codes for a nucleic acid. Codes for a nucleic acid, because you've got to remember that the mRNA, the tRNA and the rRNA are all also coded by the DNA. So the latest definition for gene is that number one codes for a polypeptide or a protein, and number two codes for a nucleic acid. So you see, as you know, mRNAs of course result in the formation of proteins, but then tRNAs, where do they come from? They are also coded by the DNA. Similarly, the ribosomal RNA is also coded by the DNA. So this is a very newer version of the definition of gene. Then we have the second point, and the second is the locus. Locus means position. Like we have a chromosome here, and we say this is the gene for eye color, this is the gene for blood group. So locus means the position. Where is it present? Because please remember, chromosomes are made up of base pairs, like you have A, T, G, C. So these are called BP or base pairs. So after how many base pairs do we have this gene, and how long and how many base pairs is this gene? So those are the important questions that we need to understand. Then we look at the word allele, and we look at the word dominant and recessive. So we've got three new terminologies. So allele is the alternative form of a gene. Alternative form of a gene. Now, for instance, like here, I keep on changing colors. So I have how many colors? I have 1, 2, 3, 4, 5 colors, as you can see on the top. So I have 5. If I have a pen, I have 5 different colored pens. Now, important thing is allele is the alternative form. So if you look at the example which I like giving you is blood group. Now, there are 4 blood groups. Like for instance, we say there could be a person with blood group A, there could be a person with blood group B, or a person with blood group A, B or O. So there are 4 blood groups. But when we look at and analyze all the genes for it, we find that there are 3 alleles, and the Institute of Biology decides who's going to and what letters are we going to give it. So it's IA, IB and IO. The reason is because it's a codominant phenomena, so we write something raised to the power something. So we actually have 3 alleles for blood group. Number 1, number 2 and number 3. But the different combinations are very variable. Like for blood group A, we can have IA. Now this would be homozygous. And because we are writing a capital letter, so it would be dominant. Or it could be IA, and there is a recessive one which is IO. Similarly, for blood group B, we have IB, O we have IBIO, right? For O group there's only one possible phenomena, because no other one, there's only one possibility. Similarly for AB there is also one possibility. So if it's IA or IB, now this would be. Now when we give this letters, like this, this, and we write this in letters, then it's called a genotype. But if we give it names like the person has blood group A or a person has blood group B, now these are actually things we can test, and we can actually give it, we can do a blood test and we can find out. So we say these four, blood group A or blood group B, this is now will be called a phenotype. And I tell you to remember it by saying a pH, phenotype, pH for physical features like a person has brown eyes, or a person has black hair or blonde hair, or the person is blood group A. So phenotype, physical features. Genotype is always expressed by letters, and either we use two letters or we use one letters. We're going to just discuss that. If it's a phenomena of codominance, then we use two letters, like I said, I raised to the power A and I raised to the power B. So this is codominant phenomena. So genotype is always we use letters. Phenotype is the physical features, and we will say the person has blue eyes or brown eyes. So brown eyes or blue eyes is the phenotype. Genotype will always be expressed as letters, and we often ask you in questions what is the genotype, and of course there's some information given in the question and you are supposed to figure that out. That will be the end of this video, and then we continue this in the next video on inherited change. Thank you.